Hello, everyone, and greetings from Garching, Germany, or good afternoon, good morning to all of you joining from Europe, North America, or any other parts of the world. I am Laila El-Hash, Marketing Manager at MSAID, and I'm happy to welcome all of you to our second webinar of the year and our first time featuring guest speakers. Please note that this webinar will be recorded for documentation purposes. And today we have an exciting agenda lined up for today's webinar. Our CEO, Martin Frejno, will kick off the session and provide a brief introduction to CHIMERYS and our intelligent search algorithm, followed by our two guest speakers, Rupert and Manuel Matzinger. Both are postdoctoral fellows at the Research Institute of Molecular Pathology in Vienna, and they're currently focused on method development in single-cell proteomics. Rupert and Manuel will delve into the experiences and collaborative advancement in the field. They will also share their findings from their new publication, which it was also in the invite of the webinar, which includes insights into the utilization of CHIMERYS technology. And towards the end of the session, they will be addressing all of your questions. And I guess that being said, I will pass the virtual stage first to Martin. He will also provide first the introduction of CHIMERYS. And we encourage all of you to participate by asking questions and also sharing your thoughts in the Q&A session throughout the presentation. We will also do our best to answer your questions during the Q&A session that follows. So Martin, I can see your screen. I will just stop here. Yes, awesome.
Thanks, Laila, for the introduction. Thank you, everyone, for participating. Let's dive straight into today's topic, and that is analyzing wide window acquisition data with CHIMERYS. So Laila said that we welcome you all from Garching. Just a couple more words who we actually are. MSAID stands for Aiding Mass Spectrometry with AI. MSAID is the company behind CHIMERYS, as well as INFERIS and INFERIS rescoring, some of which are tools that you might have used in the context of PD already, either PD 2.5, 3.0, or now 3.1 since last year. We develop multiple tools. The three that I mentioned are listed here, but today's seminar is solely focusing on CHIMERYS, which is our intelligent search algorithm that makes use of predictions of fragment ion intensities as well as retention times that come from INFERIS, which is our machine learning model. So without further ado, dive straight into why you would even care about CHIMERYS and why it is even called CHIMERYS. So this is one slide that I always show when I want to give a brief introduction into the problem of chimeric spectra. Some of you might have heard about that problem already, and it's very ubiquitous in all kinds of mass spectrometry data. So what is a chimeric spectrum? Here we have a plot that visualizes the number of precursors from a spectral library containing 400,000 precursors as a function of precursor MZ and retention time. So if we now randomly place either DDA isolation windows, wide window acquisition isolation windows, or DIA isolation windows in this map of precursors, in the majority of all cases, you will identify, you will basically pick more than a single precursor for fragmentation and hence generate a chimeric spectrum, the definition of which is that it consists of signals coming from multiple different peptides. The problem is not really new. It has been around for quite some time, and several software packages tackle the problem in various different ways. And CHIMERYS is one way of tackling this problem with a prediction-first approach. But before we dive into how CHIMERYS deconvolutes chimeric spectra, just a bit of background on the difference between the three acquisition methods that I mentioned here. So DDA, wide window acquisition, and DIA. This is from a beautiful paper that also took a look at wide window acquisition, and it visualizes the key problem. So when you are looking at an experimental MS1 scan, sort of schematically depicted here in the top left corner, and you were to analyze that precursors present in that scan in the data-dependent fashion, then the precursors you would try to identify or pick for isolation and fragmentation are delineated here in colors. And you see that the isolation windows are somewhat tight around the precursor, and typically search engines search only 20 ppm around the precursor. And that is, of course, in stark contrast to data-independent acquisition, where the entire MS1 mask range gets covered by isolation windows that are next to one another, usually much wider than the typical DDA isolation windows, and typically also isolate and fragment multiple precursors at the same time. Wide window acquisition now is a bit of an in-between the two different approaches, where instead of isolating now the blue precursor here with a narrow isolation window, you instead isolate it with a wider isolation window and intentionally also co-isolated with these other two precursors that are next to it. Right. So why would you even do that? Well, you would be doing that only if you have a search engine that considers for candidates for the scan that results from that blue isolation window all peptide candidates that fall into the entire isolation window. Typically, if you're working with MaxQuant, for example, even if your isolation window is this wide, typically MaxQuant will only focus around on 20 ppm around that precursor, which is the center of the isolation window. CHIMERYS doesn't do that. CHIMERYS scores the entire isolation window and formulates the identification process from experimental spectra, experimental MS2 spectra, that is, as an optimization problem. What does that mean? It means that CHIMERYS tries to explain as much experimental intensity with as few peptides as possible. So what does that mean? We have here an example MS2 spectrum with the experimental intensities below the x-axis. And if you were to take, say, MaxQuant and try to analyze this particular MS2 spectrum, MaxQuant would come up with a peptide identification. And if you use MaxQuant's second peptide approach, it may even report a second identification for that scan. However, CHIMERYS doesn't stop there. CHIMERYS can identify a third and even a fourth peptide in this experimental spectrum. And if you pay attention to the intensities in this scan, you will see that the predicted intensities here above the x-axis align very nicely with the experimental intensities below the x-axis. So we try to explain as much experimental intensity as possible with as few peptides as possible. You see the majority of the high abundant peaks are actually identified or matched to one of these four peptides. If you now also consider retention time as an additional prior, or our retention time prediction, you see that these four peptides are supposed to co-elude with one another. And if you look in the MS1 isolation window, you will see that all four precursors are there. However, they are not clustered around the center, which we marked here with a dark blue line. This is 20 ppm around the center of the isolation window. So this particular precursor was actually picked. So MaxQuant would have identified this one, but it would have not identified the green one or the light blue one here. CHIMERYS is able to do so because it considers all candidates in the isolation window. As a result, if you increase the isolation window width in a typical experiment measuring a 1-hour HeLa full proteome here on an Orbitrap eclipse, you see that as you increase the isolation window width, you increase chimericity in the scans. And because CHIMERYS can deconvolute these spectra, we nearly linearly increase the number of PSMs as the isolation window increases, even though we don't increase the number of scans, right? So it's the same machine, it performs the same number of scans in the same amount of time, but we nearly linearly increase the number of PSMs, but only if we are actually using CHIMERYS, because CHIMERYS efficiently deconvolutes chimeric spectra. Sequenced with precursor detector different approach to chimeric spectra deconvolution also increases in the number of PSMs that it reports as the isolation window becomes wider, but it's far less sensitive. But this sensitivity, of course, needs to come also with proper FDR control, and that we ensure in CHIMERYS. Usually, when we want to measure this empirically, we use entrapment experiments. If you have questions to entrapment experiments, we can save them until the end of the presentation. I'm more than happy to explain this to you. All that you need to know is in an entrapment experiment, what you end up with is a plot. On the x-axis, you have the target decoy FDR. On the y-axis, you have an empirical measurement of FDR. And the empirical measurement and the reported FDR in an ideal world should correlate along the diagonal here, which is in dark black dashed line. So when you use Sequus HT with precursor detector, the empirical FDR is actually slightly higher and becomes even higher as the isolation window becomes wider. Meaning that sequenced HT with precursor detector doesn't control its FDR very well. But if you're looking at CHIMERYS, on the other hand, you see that as we increase the isolation window width, there's virtually no difference between these FDR curves, and they are very close to the diagonal, meaning that even with very wide isolation windows, as wide as 8 Thomson, we can still properly control FDR. The workflow of CHIMERYS consists of several different steps. We need to recalibrate our models based on an initial search, and error estimation later on is performed using Mockapot. But given the sort of generic way of reformulating the process of identifying PSMs from spectra as an optimization problem, CHIMERYS naturally extends from DDA data to DIA and PRM data. So we can effectively use the same algorithm to analyze any type of acquisition method. So that is a very key feature. However, we focus here on wide window acquisition. So I'll leave it at that. I'll just close here with a couple takeaways. Wide window acquisition, as I told you, intentionally generates chimeric spectra that CHIMERYS then later deconvolutes and identifies all peptides that contribute to the chimeric spectra. CHIMERYS needs to consider all candidates in the entire isolation window to do so, and it does. That actually results in a substantially increased search space. However, even with this bigger search space, given the entrapment plots that I showed you earlier, we can still properly control FDR. And as such, even in these very wide isolation windows, FDR is controlled, and hence we can natively extend the CHIMERYS logic to PRM and DIA data. So on the back of ML, I think we can say that we can predict a bright future for machine learning and CHIMERYS in proteomics. And with that, just briefly, thanks to the MSAID team who makes all of this possible, as well as our collaborators at Thermo Fisher. And with that, I hand over to our guest speakers. I'm very excited to hear about their research.
Thank you very much, Martin, for your valuable insights on CHIMERYS and also an overview of the wide window acquisition application. So yes, that being said, I will now hand over the virtual stage to Manuel and Rupert, who they will be leading us through an insightful exploration of wide window acquisition and also CHIMERYS technology. And yeah, also they will share all their insights based on their recent application. So warm welcome again to both of you. And yes, I can see your screen. Then the stage is yours. Thank you very much.
Thank you very much for this very kind introduction, and it's a pleasure for us to introduce our insights to what we did with wide window acquisition in connection with CHIMERYS. Let me first start by introducing, if this works now, by introducing our group. So we are both part of the technology hub at the IMP in Vienna, and our dedication is to more or less purely to method development. And we have a strong focus in single-cell proteomic development. We just very recently got a grant worth 2.5 million euros that allows us to buy new state-of-the-art equipment to further boost this research. Besides single-cell and low-input proteomics, we also do some work in protein cross-linking, mass spectrometry, as well as precision medicine. So when we talk about development, then of course, we also have to think about the ideal proteomic workflow. And this ideal workflow includes, of course, steps starting with sample preparation and ending with an actual biological question to answer. So we will focus today on those two parts, chromatographic separation and data acquisition, and about the needs that we need to fulfill there. So what do we need for chromatographic separation? We want to have, of course, a high throughput. This is especially important when we think about later single-cell proteomic applications where we need to analyze hundreds to thousands of cells. But we still want to maintain a high separation power and a high reproducibility. And of course, we want to have a long lifetime of our columns. So for the columns, we will focus later on what the differences are between the packed bed and the micropillar columns that we tested. And for data acquisition, of course, we also have a lot of needs, a lot of things we want to have, including quantitative accuracy, again, a high speed. So we need fast detectors, high sensitivity, sufficient resolution, and a high data completeness. And to deal with that, we of course also deal with different acquisition strategies, and one of these is wide window acquisition, on which we will focus today. So I will start to focus on the liquid chromatography part. As you can see in the illustration here, of course, the peak width also determines the actual signal intensity. This means the sharper our peaks, the higher the intensity. And when again thinking on low input samples, these high intensities are very important to have enough sensitivity to actually detect peptides of interest. Furthermore, and this is important for matching between runs and hence for high data completeness in the end, we want to have a high reproducibility between analytical runs. So the retention time shift should be as small as possible to be able to compare from one to one. So how can we actually achieve a high throughput gradient? The trick that we apply also in our recent publication is to speed up the flow rate doing loading, doing washing and column equilibration to up to 1 microliter or sometimes even higher flow rates per minute. And with this, of course, do this as fast as possible. However, during the actual active gradient, we go down with the flow rate to improve sensitivity. And this actual gradient here, of course, can vary a lot in length, as you will see on the subsequent slides. To load the sample on the actual analytical column, different options are available. Either you can inject directly or use some kind of tips for cleanup or trapping columns, as we do in most of our single-cell workflows. Two different general column types were tested in our labs, either packed bed columns, which are the standard columns, or superficially porous micropillar columns. When using a dual column layout, you can further speed up the actual throughput. However, this was not done in the work that we showed to you today. So we just talked about the different column types. And the classical packed bed column, as most of you, I'm sure, will know, consists of spherical beads that are porous and that are, of course, not perfectly homogeneous in size. And this means that different molecules of the same analyte will have a slightly different path through this column that has a slightly different length. And this, in the end, eventually leads to peak broadening. So the idea now of these micropillar array columns that we use in our recent publication is to have this perfectly ordered, highly symmetrical and equally sized pillars on a wafer, a silicon wafer. And now all the different molecules of the same analyte would move at the same speed through this column because they have to pass the same distance. In conclusion, we should end up with sharper peaks and a better resolution in chromatographic separation. And this is especially pronounced for longer gradients and higher inputs, where we saw already in previous work that we still maintain very sharp peaks also for 4-hour gradients or something else that is very long. Another feature of the MIPAC columns is that they are grounded. And as you can see in this example data here on methionine oxidation, the degree of oxidation is significantly reduced to roughly half of the level that we see on a PEPMAP column in comparison. And this further reduces the sample complexity that we have to deal in the end in the mass spec. And it also increases the intensity of the non-oxidized peptides. So again, something important for low input proteomics. With this, let's go to the first real data from our publication that is introduced today. So here we compare different column types. All of them, but the last one, are packed bed columns. So this is the PEPMAP column that it was the standard column before in our lab, or the NanoEs from Waters, or the Aurora column from Ion Optics that is very commonly used in single-cell proteomics nowadays, and the newest generation of the 5.5 centimeter high throughput MIPAC column. You can see that both on protein level and on peptide level, the numbers increased substantially and significantly, and especially on peptide level, up to 50% compared to the PEPMAP column. So we're very happy with these results, and of course continued to use this column type for our actual workflow. So what did we do? We generated a highly complex sample to really stress our system, consisting of 3 different proteomes: human, yeast, and E. coli. Then did the actual separation on the MIPAC column, tested different column lengths here. All the data was acquired on an Orbitrap Exploris 480 using wide window acquisition, in most cases at an isolation window of 4. And the data was then analyzed using CHIMERYS in Protein Discoverer. So you can see here 2 different gradient lengths and different injection amounts ranging from 10 to 400 nanograms. And as expected, I'd say more input leads to more protein IDs. That's nothing special. And you can also see that the middle bar, which correlates to the 50 centimeter column, always is a bit higher. So this 50 centimeter column performed the best in our hands in terms of total protein IDs. And this was independent of the actual amount of peptides injected and independent of the gradient length used. To make it a bit more complex, let's take a look to the overall picture here, comparing all gradients and all columns that we tested down to 5 centimeters, 5 minutes on the 5 centimeter column. So this short column was the only one that we used also for very short gradients, as we can do the loading there faster. So it made more sense to use the short column for this. Again, you can of course see the correlation of protein IDs to the actual input amount. Of interest, you can also see here when focusing on the low input, so 10 nanograms, and the 5 centimeter column, that we can actually substantially shorten the gradient length down to somewhere at 15 minutes without losing coverage of the actual proteome. And this was, of course, very engaging for us because throughput, all this matters. Mass spec time is limiting, so we want to have as short gradients as possible. If, however, maximum numbers matters, then again, as mentioned before, the 50 centimeter column and slightly longer gradients performed best in our hands. When talking about throughput, we of course have to think about run-to-run times. So for an active gradient of 30 minutes, the actual run-to-run time varies from roughly 15 minutes to more than 90 minutes, depending on the column. And these differences are visible because of the different speed of loading and washing and equilibration of the column. And when we then calculate the number of protein IDs that we can actually identify per minute, the 5.5 centimeter column outperforms all the others, thanks to this reduced run-to-run time. And the shorter your actual gradient, the higher this actual protein ID per minute number goes. So we have here now a very high throughput method, even for label-free proteomics, with more than 250 proteins per minute identified. And we tried to, we calculated the actual samples per day that we need for each method. And you can see that for this shortest one, we are close to 100 samples a day, which is already something usable for single-cell proteomics, where, as mentioned before, you want to analyze hundreds of cells per day, per experiment, not per day, obviously, in this case. I would like to mention that here our maximum flow rate for loading and washing was 2.5 microliter per minute at a maximum pressure of only 300 bars. This means, although we performed all our runs on the Vanquish Neo system, you can also use other chromatographic systems that do not support high pressures as the Vanquish Neo, and it will still work the same way. With this, we have a perfect setup now for the chromatography, and I would like to hand over to Rupert for the data acquisition part.
Thank you very much, Manuel. So data acquisition and data analysis in CHIMERYS goes hand in hand. So I will present the next slides on this. So I can actually skip that. So the classical single peptide focused approach Martin has talked about already. So this is something that is nice to have, but it's slow, and in reality we have rather co-isolation chimeric spectra, which is why actually I like to work with CHIMERYS. So in our hands, we're using CHIMERYS 1.0, still on Protein Discover 3.0, in combination with Percolator and IMP UpQuant, which was developed actually in-house. So when we compare here some triple proteome data that was recorded on an Explorers 480, we tried with an isolation width of 1 and isolation width of 4, and analyzed both with MS Amanda 2.0 and CHIMERYS. I have to say, though, that we used MS Amanda without the second peptide option. And you can see already at an isolation width of only 1 m/z, we are a lot more successful to identify peptides. So on average, we identify twice as many peptides, or twice as many PSMs as we can with MS Amanda, and this results already in dramatic increase in peptide IDs, and also significant but slightly less pronounced increase in protein IDs, and the same becomes even more pronounced when we go to the actual wide window acquisition. So what is also very encouraging to see is that CHIMERYS does not identify completely different stuff than MS Amanda is doing, but it identifies most of the proteins that MS Amanda identified, plus quite some additional ones. And it does so, as Martin showed you already, at a very well-controlled FDR. So even at 0.1% FDR, this was, as Martin mentioned, also an entrapment experiment, where we had an additional decoy database that we searched against that was shuffled. And as MS Amanda did, CHIMERYS very well controlled the FDR also. What we can actually also see is that it very much depends how wide you should set your isolation width. That depends on the sample type and also on the sample amount that you inject. Here you can see with 400 nanograms of HeLa, we actually have an ideal isolation width for maximum protein IDs of 4 m/c. It does 3 or 5 would work pretty well also, as you see. And the lower you go, the wider you can actually go or should go even with your isolation window, because as there's less stuff that elutes at the same time, the complexity is reduced, and to still keep the same complexity per isolation window or per fragmentation spectrum, you can actually and should widen the isolation width, ending up at around an ideal isolation width in our hands for 12 m/c for 250 picogram of HeLa, which we also then used for single cells. What you see here is comparison of low input material. So we had a look whether data dependent or what the difference would be in the results for data dependent and wide window acquisition, both analyzed with CHIMERYS actually. And you can see, even though the numbers, the absolute numbers are not that staggering and exciting these days anymore, that wide window acquisition performs a lot better than data dependent acquisition. Also, when we look at the precision, actually, we see that wide window acquisition performs equally well, or potentially even a little bit better in some cases than data-dependent acquisition. Particularly when we use match between runs, this is a lot more pronounced that the wide window acquisition performs better. I'm not entirely sure why that is, but we were happy to see it nonetheless. Also, when looking at the double proteome sample, so basically we had yeast and human combined, so always a total amount of 200 nanograms, with different combinations of human and yeast. So one sample was 200 nanogram human only, then 150, 150, and corresponding up to 200 nanograms always with yeast. And what you can see here is that the median of the signals should be as close to the colored lines as possible, and we see that we have both with isolation with 1 and isolation with 4 very good accuracies, quantitative accuracies for the relative quantification actually. So with this actually, we were very happy to see that the wide window acquisition and CHIMERYS workflow did a pretty good job. And we went now to apply it to a project with our collaborators from the Gregor Mendel Institute. Funnily enough, they normally work with plants, but this time they actually had a project in mice. So they were interested in novel interaction partners of SMARCA5, which is a chromatin remodeler. And for that, they actually tagged SMARCA5 with a flag tag in embryonic stem cells, isolated the nuclei, and did a pull down of the SMARCA5 with potential interaction partners, and then submitted the samples to us. And so what we did, we had 3 replicates for the control sample, where SMARCA5 was not tagged and would not bind to the beads, and 3 samples with the tagged SMARCA5 and the potential interaction partners. And we once analyzed them with a 50 centimeter packed bed column at an isolation width of 1. Another time we analyzed it with an isolation width of 1 and 110 centimeters micropack column, and a third time with a 110 centimeter column and an isolation width of 4. And as you can see, we are able to continuously improve the number of protein IDs to nearly to over twice the number of proteins. for the most advanced workflow. So and this actually also allowed us to identify a lot more potential interactors. So what you see here is two volcano plots. On the left-hand side, what would be more abundant in the controls, and on the right-hand side, what is more abundant in the SMARCA5 flag samples. So what is actually corresponding then to potential interaction partners. The big dashed lines here indicate a false discovery rate of 5%, and the finely dashed lines of 1%. And as you can see in the standard workflow with MS-Amanda 2.0, an isolation width of 1 and the 50 centimeter packed bed column, we ended up with around 26 potential interactors next to our bait protein SMARCA5, which is always good to find, of course, in a co-IP. But in contrast to using CHIMERYS an isolation width of 4 and the 110 centimeter column, we could nearly double this number of potential interactors, which was very exciting for us to see. But what about these interactors? Is this just some random stuff that we find? And indeed, no, it is not. So what you see here on the left-hand side is again the standard workflow that we used before. There we had from the 26 interactors. The ones that were identified uniquely in this workflow were actually all not known as SMARCA5 interactors, or rather ribosomal proteins that we were pulling out here, so not really of interest. The ones that actually overlapped with our advanced workflow, 61 of them actually were previously known interactors. So this also gave us confidence in the sense that we find some interactors that actually make sense. And then we found 32 unique ones in our advanced workflow, of which 19% were also known. And this is, of course, lower than the number than for the common ones, but we also want to find new stuff, so kind of makes sense. And amongst these previously unknown interactors was ARID1A, which is another chromatin remodeler that had previously not been associated to interact directly with SMARCA5, but it was very exciting for us to see because it fulfills key roles in neurodevelopmental and malignant disorders and could therefore be very interesting to follow up on this potential interaction. With this, we also successfully applied this to biological projects. So we come full circle and actually fulfill the proteomic workflow, maybe not yet, but getting there step by step. And to sum up already, I hope that Manuel and I could show you that our advanced workflow consisting of the micropillar array chromatography, wide window acquisition, CHIMERYS really does a good job, which leads to improved peptide and protein IDs over typical data-dependent acquisition. We have a good precision and accuracy for our workflow with less than 7% medium CV for 250 picogram and 10 nanogram kiloruns with match between runs. And also what we see that the median fold changes that we expect for our double proteome mixes of yeast and human deviated less than 10%, which was also very nice to see. And for this specific co-immunoprecipitation project, we're able to identify more than 90% more potential interactors. And with this, I'm already at the end. We thank, of course, the organizers, MSAID, to invite us here. And of course, I thank Manuel a lot as well for this really nice collaborative work that we were able to present. And of course, a lot of other people from Thermo, and of course, you for your attention. We'll be happy to take any questions now.
Thank you very much, Manuel, and also thank you, Rupert, for the presentation. We are very happy with the result you achieved with CHIMERYS. I will also give a few seconds so that you can write your questions.
I'm just going to start until people put in a couple of questions. Thank you very much. Hi, Manuel. Hi, Rupert. Thank you for joining us, and thank you for presenting your work. And obviously, congratulations on getting this published. When you showed initially the dilution rows where you determined the optimal isolation window, did you guys play with injection times here as well? Because I recently have seen crazy long injection times with very, very wide isolation windows for single-cell proteomics applications using chimeras. And I was just wondering what your thoughts and what your experiences here were.
Maybe I'll pass the question on to you, Manuel, since you did the actual experiment with the isolation widths.
So it's a very good point and a very good question. In that case, however, we did not play around with the injection time. It was always the same, because otherwise this comparison would have been too complex, right? Because if we have 10 different injection times and 10 different amounts and 5 different columns, yeah. But I fully agree, especially for these super low inputs of 250 picograms and less or single cells. We have tremendously long injection times, sometimes above 100 milliseconds, and also very high isolation windows. And this, of course, makes sense to have enough signal intensity in the end. Yeah.
Thanks. And I guess we have one question from the chat. Oh, sorry, I'll just quick
If you
continue
want,
while I'm
you
still
can go
talking.
ahead.
Yeah,
Yes, of course.
So there's a question concerning on the different columns and concerning the levels of oxygenate methionine. And the attendee thought that the oxidation occurs before the LCMS experiment, and asks the question to expand on whether the methionine oxidation is severely occurring on one material.
Yeah, so that's of course correct. The methionine oxidation also occurs before we do the actual separation of the column. So whenever it's in contact to air, basically, it's also in contact to oxygen, so it can oxidize. But there is also on-column oxidation, and this on-column oxidation is reduced by grounding the column. And that's why we see this difference in level of either 40% oxidized methanines or 70%, depending on the column type. So the trick is not the material of the column itself, but it's the grounding.
If I might add here, for other columns, this grounding would also actually make sense, but the columns are not so sensitive to static charging then of the column, so that's why it's really a must for micropack columns, and probably that's why you automatically then see the number of oxidations drop. It would be interesting to see if the same would happen if one would ground the 50 centimeter PEPMAP column, but I would expect a similar result then.
Awesome, thank you very much. I see the other one concerning chimeras, you already answered. And also we have another one. Have you ever comparing with and without FAMES Pro in terms of IDs?
Yeah, typically we see that it's always a question what do you want to analyze, because if you want to go for a lot of material with long gradients, then FAMES and different different CVs particularly make sense. For a single cell, and if you want to go to shorter and quicker runs, then you should go for a single CV. And then also it depends if you want to have, for classical proteomics, I would actually nearly always advise to use FAMES. For stuff that is maybe not so regularly and widespread used, like cross-linking proteomics, sometimes I guess it can be an advantage to use FAMES or not use FAMES at times. And for immunopeptidomics, I've not tested it myself neither, so it really depends what you're doing, I would say.
Yeah, but in our lab we actually always use it, and the number of compensation voltages we use is really dependent on the gradient length. So for longer gradients, as Rupert mentioned, we use multiple ones, and for short ones, only one to save time.
Another question here is, what source is being used with the micropack column, easy source or flex source? So we use the easy source.
Thank you very much. Martin, please.
I have a more of a meta question, to be honest. you certainly look at many different spectra, especially when you do your validation of interesting targets and co-IPs, for example, and you're very well versed in terms of manual spectra interpretation, I presume. So the question is, how many matched peaks are enough for you to confidently call an identification?
If I might start first,
Yes,
Manuel, that very much depends for me on what the task at hand is. If you want to generate as many potential interactors as you can, I'm super relaxed. If I want to be absolutely sure that this peptide is in there and this protein is in there, I would just really be very stringent. So to me, it very much depends on what I want to do.
So what would be the minimum?
Wow.
In a relaxed way, in a relaxed fashion, the minimum.
I think with the gut feeling, I would now say 6 or 5 maybe, but then it also depends if I want to go for single cell and I really want to go up with a number of IDs, knowing that the hits might be a bit doubtful, then I would potentially even go lower, particularly also if the retention time fits very nicely and the intake mass fits very nicely. So yeah, it's very, very hard for me to give like a concrete number there.
Then maybe a brief follow-up question going in the direction of sort of when you're doing DIA experiments, for example, when you say you want a minimum of 5 peaks present, how many data points across a chromatographic peak do you deem sufficient to call an ID in DIA proteomics? I'm trying to get a feeling, like also the audience, like if you could tell me what you think is a good value there, that would help substantially.
This is something we discussed really a lot, I think, in group meetings. And I think that's also discussed in the single cell community. And I think you have to differentiate between IDs and quant IDs, right? So if you just report a number of identified proteins, then for single cells, sometimes 1 data point per peak is even enough, right?
So correlations on
Pardon?
on 1 data point across the peak.
Yeah, exactly. So there's no correlation possible. There's no quantification possible, right? So that's why I said before, it depends on what you want to see, right? But this is sometimes, of course, reported to have a very high number in a graph or whatever that looks fancy. And then if it comes to quantification, things change, of course. And in the preprint that we when did we put out the Astral paper? I don't know, 3 weeks ago or 4 weeks ago, we published a preprint using the Astral and DIA, and there we have 5 to 6 data points per illusion peak.
How do you measure this?
We actually took the number from SpectraNote that is reported there. So I think it's at full width half maximum. And if you would go to the baseline more or less at 10% height, you would need to multiply this number by 1.7, right? But this is also a discussion point, indeed, how to measure. But we decided to use the default number that is given by the software because by that it's easy to reproduce by everyone, easy to compare to other labs. And here also on Twitter, we got responses to our preprint asking, okay, is this enough for quantification, right? And at least in that case and in our hands, it was enough because we also did 2 proteome mixtures with different ratios between yeast and human proteomes. And the quantification was both accurate and precise, at least for the fraction of, I'd say, the top 50% abundant proteins in this single cell-like mixture. So it was a total amount of 250 picograms. And if it goes to the very low abundant proteins, right, then the variation gets crazy, right? We have then CHIMERYs of 40% or more. So this is also important to consider. The lower abundant, the trickier it is. And median of 5 data points per peak does not mean this is true for all peaks, for all IDs, right? So yeah, tricky question.
Yeah, but thank you for your insight. We think about these things a lot at MSAID and do our own analyses, and yeah, so there's lots of interesting stuff to see and find, and we'll share some of these analyses also at ASMS.
Great, I'm looking forward to it.
Awesome. So we have another question. In addition to the max injection time mentioned by Daniel, when you widen the isolation window, do you also increase the AGC target value? What is a good strategy to change the AGC target value?
Oh, this is a good question.
I think we actually didn't change that.
No,
right?
I think we kept it
Constant.
Yeah. But this is something that I don't remember from the top of my head. There, I would really need to go into the raw files, and there's so many raw files in submitted to Pride. I think it's 2 or 3 or 250 raw files. So I would need to check. There's an overview, a supplementary file there where the AGCs is included. So I'm sorry that I don't know it from the top of my head, but please have a look there.
Awesome. Thank you very much. And I guess the last one, again, from the data acquisition aspect, how about the margin of the dynamic exclusion? The default is plus and minus 10 ppm on my Lumos. Even opening the isolation window, should I use the same default? Which means the ones already isolated and the one way will be chosen again. At that time, the previously selected precursor and the MZ. One way from the new selected precursor is included again. Is setting the half of isolation window on plus side and minus side good idea?
Yeah.
If I might start first, Manu, I think we, if I remember correctly, we tried to play around with the dynamic exclusion, and we were a bit surprised to see when we set it to a wider width that we actually lost IDs again, or it did not improve to the extent that we had hoped for. So yeah, I'm not sure why that actually is. But in our hands, widening the dynamic exclusion did not improve things.
Awesome. Thank you very much. I guess if we don't have any more questions, I would like to thank you very much. Thanks to you, Manuel, and also Rupert. Thank you, Martin and Daniel as well. We hope that you all gain valuable insight into the application of CHIMERYS through wide window acquisition and also data analysis. If you haven't tried CHIMERYS, I will just share my screen, and then you will have access to a QR code where you can easily sign up a form and you can request our demo. And yes, thank you very much again. And we will ensure the recording is available on our YouTube channel for your convenience. Stay tuned for more exciting updates from MSAID. Thank you again and have a wonderful day.
Thank you. Bye.
Thank you,
Thank
guys.
you.
Bye-bye.
Greetings to Vienna. Bye-bye.
Bye-bye.
See you soon.
Bye.